Assalamualaikum and good day everyone. I hope you're all ready to learn and discuss brain imaging and neuropsychology. I hope that at the end of this module you will be able to define and describe type of brain imaging modalities and its application, and describe the integrations of brain imaging and neuropsychology. For those who already took GCN 508 Fundamental in Neuroimaging will found that part of this topic is not new and already been discussed last semester. Although this topic is not new for some of you, I hope you are always enthusiastic in reviewing this topic to better understand and remember it. Before we proceed, I would like to inform you all that I separate the discussion for this topic of brain imaging and neuropsychology into 4 modules and 1 tutorial. Module 1 is the Introduction to Brain Imaging. Module 2 is the Structural Brain Imaging Modalities. Module 3 is the Functional Brain Imaging Modalities. Module 4 is Introduction to Neuropsychology, and then proceed with the tutorial. For tutorial, the student will be given a journal article that integrating neuropsychology assessment and brain imaging to be discussed and shared in the class. What is brain imaging? We should thank previous and recent scientists who have provided a powerful method in exploring structural and functional of the brain. This is one of the benefits of innovation in physics and computing technology. With the emergence of imaging technology, we can explore the details of how a single neuron might function and how a billion neurons integrating or communicating with one another and process information in the human brain. Brain imaging technologies allow scientists or researchers to look inside the brain to see the effects of injuries, diseases, drugs, and other chemicals, and etc. Brain imaging, also known as neuroimaging, falls into 2 broad categories. The first category is structural imaging. Structural imaging provides images of the structure or anatomy of the nervous system. For your information, the cerebral cortex is a highly folded sheet of gray matter that lies inside the cerebrospinal fluid and surrounds a core of white matter. This type of imaging helps to analyze cortical structures and in the diagnosis of brain injury and the diagnosis of certain diseases. Furthermore, this technique also can answer how the structure of the brain, which is crucial when discussing cognitive development, changes with age, changes with rehabilitation method apply, changes with treatment. Next, the second category is functional imaging. Functional imaging provides images of metabolic changes or brain activity while patients or participants complete tasks such as reading, memorizing, solving mathematics problems, doing motor activities like finger tapping, auditory sounds, visual tasks, and many more. The brain area or areas that are involved with completing or responding to these tasks light up. Functional imaging measures stimulus-related changes in blood flow or metabolic changes. It will show the parts of the brain involved with each type of task. Moreover, this technique can be used to explore functional developments of the cognitive process, particularly for memory, decision-making, speech and language, and others, which are important for human cognition. This type of imaging can also be used for research purposes in various areas, for example, for neurological, cognitive neurosciences, cognitive psychology, and computational neuroscience.
Here is a brief historical overview of major events associated with the development of human brain imaging. In 1924, Berger succeeded in recording the first human electroencephalogram, or EEG. His first paper demonstrated the technique for recording the electrical activity of the human brain from the surface of the head, published in 1929, which are 5 years after his first human brain electrical activity recorded. In December 1946, Edward Mills Purcell, an American physicist, discovered nuclear magnetic resonance phenomenon with his colleagues, Robert Pound and Henry Torrey. This NMR phenomenon becomes the basis of magnetic resonance imaging, one of the most important medical advances of the 20th century. In 1956, infrared imaging in medicine was introduced. Ray Lawson used infrared imaging in breast cancer patients and discovered higher skin temperature above cancer spot than a normal tissue. In 1961, James Robertson and his associates at Brookhaven National Laboratory built the first single-plane PET scan and gave a funny nickname, the H-Rincon. In 1963, brain blood flow linked with behavior. Positron emission tomography, or PET scans, work after radioisotope traces are introduced into the patient. The imaging equipment picks up gamma rays emitted as a result of isotopes decay. The system allows for functional imaging of processes throughout the body. The device is now used for research and to diagnose certain cancers, brain diseases, and heart problems. Next come SPECT in 1968 onwards. In 1968, David Cohen performed the first MEG at Massachusetts Institute of Technology. CT and MRI were introduced and developed in 1970s and 1980s. In the early 1990s, blood oxygen level dependent or BOLT contrast to map brain activity was discovered. In 1991, the first functional magnetic resonance imaging maps of human task-based activation using a visual stimulus paradigm were generated by Jack Bouvier. The development of human brain imaging keeps updated up until now.